biology at the School of Medicine. She received her PhD from Boston University and has been the recipient of a prestigious NIH Director's New Innovator Award, where her work has been focused, akin to the title, on understanding cell fate and cancer cell of origin. And I think, among other things, her laboratory work is really to understand what are the rules of cell fate, the decision process, in part to understand how to avoid harm and as well understand how cells ultimately become cancer. So, Dr. Guo, thank you.
I'm grateful for this opportunity to share with you our work, and this is the vast expanse of what we're talking today. So on one hand, we just heard that doctors take care of cancer patients, but now I would like to bring you to the very, very beginning, before the normal cell has crossed over to the dark side to become cancer. So the question that ultimately I'm asking is, what kind of normal cells give rise to cancer? So the way that I was taught cancer biology is very similar to this, that put together by Bert Vogelstein, particularly and colleagues. So during the cancer development, these normal cells sequentially acquire more and more and worse mutations, such that, I don't know whether I can have the arrow here. Okay, I don't think I can.
You have to use the mouse on your laptop.
Okay, okay. So one of these blue bars indicating the normal cells would sequentially or successively acquire worse mutations and become bad, eventually gives rise to some kind of malignancy. So in normal cases, in most cases, that the malignancy is a protracted and rare event. It could be so rare that as long as one cell goes through this process, we will see clinical cancer. But because it's a rare and a protracted event, the identity and behavior of the cancer cell of origin has only been, logic detection lives in our head. And this sometimes is also called the multiple hit model, where the full transformation requires accumulation of many genetic abnormalities. With the advent of genome sequencing, we do know now that the number of mutations observed in a wide variety of human cancers are not always equal. On the right-hand side, we can see that cancers like skin cancer, melanoma, or lung cancers, they are known to be particularly overladen with cancer mutations. But we also see cases of cancer types on the left-hand side. In particular, I draw your attention to acute lymphoblastic leukemia and acute myeloid leukemia. These are all malignancy of the hematopoietic lineage, or our blood, where the mutations are consistently to be very low. In particular, the acute lymphoblastic leukemia, we know that most of these cases are actually observed in pediatric patients. So here, in particular, in the mixed lineage leukemia rearranged malignancies, the recurrent genomic abnormality is on average only about 1.3. So that really says in these kinds of hematopoietic malignancies, perhaps the successive accumulation of mutations, that theory itself cannot explain all. Of course, being in the stem cell center, we also have to consider this hit in the stem cell model, which postulates if the stem cells, instead of one of these differentiated progenies, when they acquire mutations, because they live for a long time, and they are the ones that give rise to cancer. But my personal opinion is that this is somewhat not really a meaningful argument, because if we imagine the stem cells, this is depicting the hematopoietic stem cells. If you move many steps behind, that's going to be the germ cell mutation. but we do know even in patients with germline predisposition, most of their soma end up being normal. So what we are considering really is which somatic cells give rise to certain rare cell fate. Why are we even bothered with this crazy question? Is because we are believers that mutations themselves or combination of mutations are not sufficient to drive certain rare cell fate. This is mostly well known in the somatic cell reprogramming into induced pluripotent stem cells or iPS cell experiments. We know now the remarkable combination of those 4 transcription factors, several of them are known oncogenes. When you introduce these 4 genes or 4 transcription factors, even in every single cell of a mouse, still only a minute percentage of these somatic cells can ever become iPSCs. So we consider somewhat a similar analogy. Is it possible that the oncogenic mutations, even when they're accumulated inside of the somatic cells, they have to follow some rules or selectivities that the cell of origin, the initial somatic cells, may be somewhat restrictive. So our approach in the study of the induced pluripotency case has been a live cell, long-term live cell imaging and tracking. So I don't know whether you can see, but these yellow arrows were tracking a hematopoietic progenitor, a blood-forming progenitor. And when they're in the beginning, their blood cells, they undergo rounds of division. And I'll just fast forward and to show you by tracking these fate. Now at the end, we can generate induced pluripotent stem cells. We know these are iPSCs because their morphology, because they're pretty stereotypic reporter gene activation. And we are formulating this hypothesis is also because malignancy and the pluripotency initiation, they share many commonalities. I list a couple here, which compares the similarities between malignancy induction and pluripotency induction. On the left, under the malignancy induction, many features were put forward, by example, for Bob Weinberg and colleagues. Classically, they transform the normal cells, both mouse and human, with oncogenes like SV40, large T antigen, so on and so forth. Importantly, for example, P53 loss of function or CDK gain of function, they promote both of these kind of cell fate, and a variety of somatic cells can be converted into the target cells, which both in this case are highly proliferative, and it's a protracted rare event. So then our question becomes simplified. Does malignancy actually initiate from these normally already fast cycling cells? To address that question, we have to create a few tools first. First, we have to make sure that all the cells can share the same necessary oncogenic mutations, and then they can drive cancer development. So to do this, we first derived a knock-in oncogene model. In this model, we directed MLL-AF9 mutation. In humans, it's created by chromosome translocation. And we knocked this into the endogenous HPRT locus under the control of a tetracycline inducible promoter. And in the same construct, following an internal ribosomal entry site, following the same mRNA, there's this cell surface reporter, non-functional NGFR. And when this is crossed with ROSA26 RTTA, and when cells or mice are given doxycycline, all cells could similarly induce this oncogene. And this is confirmed by flow cytometry staining for this oncogenic construct. And we can also confirm this construct or this oncogene allele can drive bona fide acute myeloid leukemia or AML development in mice, and this is dependent on its genotype. So if you don't have the RTTA, even if you feed the mice with dox, there's no development of cancer. And in this case, if you give the right genotype but don't feed the mice with dox in the control line, all mice are not are viable. And the red lines indicate that those mice develop these acute myeloid leukemia. So we validated our cassette works, but these lethal leukemia could have happened as long as one normal cell has crossed over to the dark side, right? So our question now, okay, so we know now all cells can be made to express this oncogene leading to AML. The question next is, do all cells transform? I don't have the time to show you, but I can tell you the results here is actually very few cells, even in this model, can be transformed. Key questions is what kind of cells transform? So we start we attempted then to image malignancy initiation from normal hematopoietic progenitors from when we're normal. So we took the cells from the mouse bone marrow, carrying our oncogene cassette, and put them into these micro wells and started our microscopy to follow their division. So this is one particular well, but in reality, we imaged simultaneously hundreds of these single cells. So in this first, you can see when we started imaging, there's one cell. After a while, there's 4 progeny. After a week, these transforming cells gives rise to this highly compacted, dense colony. Because they're confined in the individual wells, what we can do is we can pluck them out and give them another round of colony plating. So in this serial colony replating assay, that is our definition of de novo transformation. And some of these cells, when you pluck out and to inject into the mice, you can indeed induce leukemia in the mice. So now we get to ask, we can watch hundreds of single hematopoietic progenitors all expressing the same oncogene, who transforms and which cells transform. So I'll walk you through how to read these graphs. So out of hundreds of these hematopoietic progenitors, specifically we're tracking GMPs, or short for granulocyte macrophage progenitors. About 20% of them, you see, look at the gray bars, about 20% of them within the first 24 hours of imaging never divided. About 40% of them divided once, 20% of them divided twice, and about 1% of them divided 3 times. So, and within this time frame, whether you give them doxycycline or not, it does not change their cycling behavior. So whatever we're measuring here is their intrinsic, inherent behavior. Now, this is their transformation potential. So clearly, this is showing with the cells that can normally divide 3 times within the first 24 hours we're looking at them, essentially 100% of them, all of them can give rise to transformed growth. So that's a nice correlation. So if under conditions when we accelerate the GMP cycle without the oncogene, can we alter the probability of malignancy? So here we use the emergency myelopoiesis model. Is under a challenge or stressed condition, the hematopoietic progenitors have to produce a vast number of cells to fight infection, for example, that we can see that our treatment induced the increase in the category, the 1% of the GMPs that can normally divide about 3 times or more. And also remarkably, now we see a subcategory of cells that can divide even 4 times within the first 24 hours. I'm not going to show you the colony count, but this is for the cells that are going into the mice. Indeed, we generate a much more aggressive kind of leukemia. The conversely, it's also true, is that if we pre-incubate these cells with the CDK4/6 inhibitor, in this case palbociclib, only 1 treatment or short dose of treatment. In the beginning of the DOX induction, we can slow the development of leukemia and in the sub cohort of these, they never develop leukemia. So with this, we started wondering, how does this oncogene transform the rapidly cycling normal cells? So this again is a hematopoietic progenitor stem and a progenitor tree, and here we see the stem cells on top. In the middle are the rapidly cycling progenitor cells, and down here is the circulating post-mitotic cells. So we thought if we can understand what target genes are being driven by this oncogene, perhaps we can get to some understanding. What's surprising, however, is that, so what we did is we took out these three kind of cells, by and large, and induced them by DOX treatment, just simply by doing an RNA-seq to ask whether or which kind of, what gene expression signature is overlapping. What's surprising is that there's barely any overlapping driven by the same oncogene. So in this plot, it's showing that there are only 5 overlapping genes, and one of them being the oncogene itself. So this is puzzling until we did the single cell RNA sequencing experiments. Again, we're comparing the minus DOX with the plus DOX, and these 3 are the classic, well-recognized or well-accepted MLL-AF9 target gene. What you can see is that there are small subset of cells, normal cells, have never seen the oncogene. They are already expressing all of these classic target genes. And it's not just that these 3 genes we handpicked, it's entirely their entire transcriptome. So this is in collaboration with Dan Burkhardt in Smita's lab. You can see that the plus DOX minus DOX condition, the data structure is essentially overlapping. So at least at this time point, this oncogene has really not altered the cellular state. So now that becomes a bit funny. Okay, so I will come back to this. So then we were wondering whether this this phenomenon we're seeing, these rapidly dividing cells to be supportive or to be responsible for initiating cancer, means anything for human cancer. And the answer is yes. So in the target data set, which annotated their MLL fusion status, we can see that this is a cyclin D1, CCND1 expression specifically predicts the poor prognosis for MLL fusion-based leukemia, but not for all the other kind of underlying leukemia. So this work is just in press now for Nature Communication. So to summarize, what I'm proposing as an alternative way of seeing what cancer really is or how it is coming to being is that rapidly cycling cells exist normally, but ordinarily they only are in existence briefly, in transient. And what this kind of oncogene does is that preserves these naturally existing fast cycling progenitor state. So what I'm suggesting is this oncogene, this kind, at least the MLL fusion oncogene, when it is expressed or when it meets these normally rapidly cycling this dividing cells, it perpetuates this state, and that creates cancer. So the overall model is that hit in the transiently fast cycling model. So we know in the hematopoietic progenitor hierarchy, the particular one in this case we studied is the GMP. They normally cycle fast. And when this sees the oncogene MLL fusion-based leukemia, this is what happens. This is a naturally existing cell state being preserved and perpetuated by this oncogene. And lastly, I would like to thank my lab. This work is done by a very talented graduate student, Yue Chen, and our collaborator, Dr. Smita Krishnaswamy, and her student, Daniel Burkhardt. And thanks for our funding support. Thank you very much.
Thank you. Questions?
Yes.
David.
So I may have missed this, but Shangqin, you actually, since you can identify the cells with cyclin B1 or getting out of G0,
Mm-hmm.
in your time-lapse experiments, can you show that those are preferentially going into a large clone compared to yourself?
So in our assay for transformation, it's almost binary. You either get a hugely compact colony or no colony at all. There's no in between.
So if again, in your original assays where you have a series of wells,
Mm.
single cells, and identify presumably by time lapse,
Right.
that one or two of those are hitting B1 and then follow them.
Mm.
Or can you show prospectively in that fashion that those are the subset that are going to go into large clone?
At the time when we were doing those experiments, we were not able to prospectively pick out whoever that's about to do that. So to address that question, we actually took the time to create a fluorescent live cell reporter of cell cycle speed just by looking at the ongoing cell cycle rate. Yes, so we do have a tool like that now, but at the time we did not. Haven't yet, ongoing, yes.
Any other questions?